Good afternoon, ladies and gentlemen. I warmly welcome all of you to the plenary session of the first day of the 14th International Research Conference at General Sir John Kothalavala Defence University. You are here with us today at the medicine session based on the theme towards a robust healthcare amidst mitigating pandemic challenges. This session will be chaired by Senior Professor Narada Warnasuriya and Senior Professor Mirani Weerasooriya. Ladies and gentlemen, I have the honor of introducing the chairpersons of this session. Professor Narada Warnasuriya is the Senior Professor of Pediatrics at General Sir John Kothalavala Defence University. He is also the Emeritus Professor of Pediatrics at the University of Sri Jayewardenepura, where he was formerly the Dean, Faculty of Medical Sciences, and Vice Chancellor. He has been a past President of the Nutrition Society of Sri Lanka, the Sri Lanka College of Pediatricians, and the Sri Lanka Medical Association. He is also a past chairman of the Committee of Vice Chancellors and Directors of Sri Lanka. In 2009, he was awarded the honor Outstanding Asian Pediatrician at the Asian Congress of Pediatrics in Shanghai, China. Professor Mirani Weerasooriya is the Senior Professor of Parasitology at the Faculty of Medicine, General Sir John Kothalavala Defence University. She served University of Rohini as the Senior Professor of Parasitology and Dean, Faculty of Graduate Studies, prior to her retirement early this year. During her 33-year-long career as a university academic, in addition to her teaching, service, and administrative commitments, was instrumental in establishing the Filariasis Research, Training, and Service Unit at the Kandy Society of Medicine Orator, and has published widely, gaining national and international recognition. She is also at present serving KDU as the MPhil PhD Program Coordinator. I cordially invite Senior Professor Narada Warnasuriya and Senior Professor Mirani Weerasooriya to commence the session.
Thank you, Chalindri, for that generous introduction. I would again like to welcome you to the plenary session in medicine of this 14th International Research Conference of the Kothalavala Defence University. As Chalindri mentioned, the theme is towards a robust healthcare amidst mitigating pandemic challenges. And we have got 4 speakers today, 2 clinician scientists, one based in Sri Lanka, one in the United Kingdom. Then we have a clinical pharmacologist with extensive experience in pharmaceutical regulation. And then finally, we have a public health physician with experience at the highest level of international health. They will be looking at, the first 2 would be looking at obviously the laboratory and the pathophysiological basis for the diagnosis and prevention and the overall public health response to this pandemic. We will have a local experience first, and then a perspective from the United Kingdom second. Then the third speaker will be actually presenting 3 case studies where pharmaceutical regulation greatly impacted policy making in this pandemic situation. And the final speaker will look ahead to a more resilient and a stronger public healthcare system after coming through this storm. So we look forward to a very productive seminar. My co-chairperson, Professor Mirani Weerasooriya, will actually introduce the 4 speakers. There will be no questions at the end of each presentation. but we will have a question and answer session right at the end, and then we will sum up the seminar. Thank you. Over to you, Mirani.
Okay, thank you, sir. Let me introduce the first speaker, that is Dr. Chandima Jeewandara. Dr. Chandima Jeewandara is a senior lecturer of the Department of Immunology and Molecular Medicine and the clinical lead, Allergy Clinic, University of Srijavadanam. He obtained his MBBS from University of Colombo in 2006 and the membership of Royal College of General Practitioners UK and PhD in Immunology from University of Srijavadanam. He also holds the specialty certificate in pediatric allergy by the European Academy of Allergy and Clinical Immunology and MSc in allergy from the Imperial College London. He has won many research awards, including SC Paul Oration on the Sri Lanka Medical Association, Presidential Awards for Medical Research in 4 occasions, and National Research Council in 2 occasions, and awards from NSF, NIH, WHO, and University of Srijavadanam. He has over 30 publications in peer-reviewed journals and is the author of many books and monographs. He has been a resource person in many national and international fora on dengue and other viral infections, allergy, and immunology. He is also the current director of the Allergy, Immunology and Cell Biology Unit and the director, National Center for Primary Care and Allergy Research, University of Srijavadanam. Over to you, Dr. Chandima Jeewandara.
Can you see my screen? And am I clear to you all?
Yes, we can see your screen.
Little louder, Chandima, little louder.
Thank you very much, madam, for your kind introduction. And it's a great pleasure to be with you to talk about through its robust healthcare and its pandemic challenges. I'll be focusing my talk on the vaccine immunity studies, and I must say that all data actually are generated locally.
I think, Chandima, we need to be a little bit more louder, closer to the mic.
The virus is simply a piece of bad news wrapped up in protein. The biologist Jean and Peter Meadow wrote in 1977. In January, scientists deciphered a piece of very bad news. Most extraordinary finding in late 2019 and early 2020 was from the Chinese scientists, where they sequenced a virus which is not known previously, causing a pneumonia outbreak. In Sri Lanka, we experienced as early as January 2020, and the first local case was detected from our laboratory on 11th of March 2020. It was a turning point in our modern era in global health. We have learned a hard lesson about the intrinsic vulnerability of our societies to a single pathogen. More than 219 million confirmed cases and more than 4.5 million deaths are recorded as of today, but this is a gross underestimate. The impact is enormous. COVID-19 has crippled our economies, livelihoods, lives, and social lives, and it has inequality was very evident, especially with regard to vaccines. So, ladies and gentlemen, let me give a brief outline to my talk. So, I'll be talking in the next 20 minutes about the virus, bit of epidemiology, and correlates of protection mainly, and the studies we have done in Sri Lanka, and the future challenges. SARS-CoV-2 is a single-stranded positive sense RNA virus, which belongs to lineage B of the genus beta coronavirus in the Coronaviridae family. It shares about 78% homology with SARS-CoV, and it has two major open reading frames named as ORF1 and ORF1A and ORF1B, and it also constitutes with four structural proteins, namely S protein, envelope protein, membranous protein, and nucleocapsid protein. Let's talk about the situation right now in Sri Lanka. As you can see, compared to the other parts of the world, like United States, United Kingdom, and Israel, Sri Lanka also experiencing a massive surge of cases. But I would like to draw your attention to the slide on your right, where we have a very bad situation. As shown here, the 7-day average death is very high in our country. As you can, I have shown in this slide also, compared to the other countries in the world, we have a very high mortality rate based on the reported number of cases. Probably this figure is high due to an underreporting. When we talk about the vaccine rollout, we started very slow, but we are catching up. We are catching up with all the other nations in the developed world. As you can see in this map, even in Southeast Asia, Sri Lanka is shining with the dark green spot. So we should be happy about our vaccine rollout, especially during the past few months. Our rollout was enormously good. The best and the safest way to control the coronavirus disease pandemic is by using vaccination to generate widespread immunity to everyone in the world. As of today, we have plenty of candidate vaccines approved by different authorities, and in Sri Lanka, we have several vaccine candidates being administered to Sri Lankans. There are a series of conventional and novel platform technologies used to develop vaccines. For example, live attenuated viruses, inactivated viral vaccines, protein subunit vaccines are the former forms. Currently, the latest platforms include mRNA vaccines, DNA vaccines, and viral vector vaccines, which are widely used with COVID-19 vaccination. All candidates in chemicals are designed to elicit an immune response to spike protein of the SARS-CoV-2. However, some emerging candidates are based on other viral proteins like membranes and membranous and nucleocapsid proteins. Although no immunological correlates of protection against SARS-CoV-2 has been identified, the major focus has been on the induction of neutralizing antibodies. And it is mainly directed towards the spike protein. Various T-cell subsets are also thought to be important in controlling the disease progress rather than preventing the infection. Infection-induced immunity provides a broader immune response with both systemic and mucosal immunity, while vaccine-induced immunity mainly focused on the systemic immunity. A beautiful study which was done, published in Nature, demonstrated in animals. When there is a passive transfer of antibodies, the rate of infection significantly diminishes with the concentration of the antibodies. There is evidence to support neutralizing antibodies as a correlate of protection in some settings, and also in certain vaccine efficacy trials done in several parts of the world. The role of T-cell based immunity has also been observed, and some vaccines can induce strong CD4 and somewhat CD8 T-cell responses. And we don't know fully what is the role of T-cells and the quantitative contribution for protection with regard to T-cells in giving protective immunity. Mucosal immunity plays a significant role in protection. As you can see in this slide, natural infection produces both mucosal immunity in the upper tract as well as IgG responses in the lower tract. But the currently used intramuscular vaccination mainly focuses on the IgG immunity. That's why with the current candidate vaccines, we don't get sterilizing immunity. We still can transmit the infection to the others. The future generation vaccines will probably focus on intranasal vaccination. The current data with the existing vaccines show that the development of mucosal immunity is not that great with the existing vaccine platforms. With that data in our mind, we designed our vaccine immunogenicity studies in Sri Lanka because at the time of registration of the vaccine, there was a huge controversy among the health and the non-medical staff about the introduction of vaccines to Sri Lanka. So we had, except for few vaccines, the vaccines we used in Sri Lanka had little data, and we thought it's important for us to have our own data, and we designed different cohorts of vaccine studies with our population in several districts. We took a blood sample prior to vaccination, and based on the vaccines they received, we took another blood sample at 1 month, and we followed them prospectively long term, and some of them are at third month point, and some of them are 6 month point, and we'll be prospectively following them up for about 1 year. So to assess the immunogenicity, we use several methods. We determine the total antibodies in those people who receive the vaccine, and also we check the neutralizing antibody levels in people who receive the vaccine with two methods: the heme agglutination test, which we have close collaboration with Professor Alan Townsend from Oxford University, and with Lymphaban from Singapore. We use the surrogate marker assay to check the neutralizing antibodies because we lack the VSL3 facilities in Sri Lanka. Apart from that, we used ex vivo and cultured T-cell assays to determine the memory T-cell responses, as well as we use the B-cell erythrocytes to determine the B-cell responses in our cohort. The first vaccine used in Sri Lanka was Covishield. Let's look at the data for Covishield. As expected, there was a very good seroconversion with both vaccines, with the vaccine in both groups who were naive at the time they received the vaccination, as well as the ones who had a previous infection. So our results showed that 93.4% were seroconverted with AstraZeneca vaccine. Further, we went on to check the neutralizing antibodies, as I mentioned earlier, using a heme agglutination test to wild type as well as variants of concerns, namely alpha and beta. We started these vaccine trials way back in February and late January, and we didn't have Delta variant circulating much in the world. But later we introduced Delta also, and we determined the Delta antibodies also in these cohorts, and we saw the antibodies developed to variants were quite significantly higher compared to the natural infection. And also they have very good levels of neutralizing antibodies following a single dose of Covishield vaccination in our cohort. The second assay we used to determine the neutralizing antibody was the SVNT, and again in this cohort also we observed there was a very good production of neutralizing antibodies in our population, and with single dose AstraZeneca vaccine it produced robust antibody responses targeting the RBD receptor binding domains of the virus, which is associated to be with protection. These are the data for our T cell and T cell studies. As you can see here, our cohort developed significantly higher responses to both S1 and S2 region of the virus, and these interestingly, the T cell responses did not correlate well with the antibody responses. So in summary, ladies and gentlemen, Covishield vaccine at day 28 produced significant levels of antibodies, and their T cell responses were quite good, and they had very good memory cell responses also. The second vaccine, Sinopharm vaccine, was introduced as early as April in Sri Lanka, and there were limited data on the immunogenicity of the Sinopharm vaccine, and there was a huge demand from the Sri Lankan authorities to produce our own immunogenicity data. As expected, more than 95% of the individuals seroconverted with after getting 2 doses of the vaccine, especially at 6 weeks. 81.25 of the individuals had neutralizing antibodies, and these levels were comparable to the natural infection. As we did for the Covishield vaccination, we checked the RBD antibodies, that is one way of checking the neutralizing antibodies to different variants, and we saw the antibodies developed against variants were significantly reduced compared to the wild type. However, they still had some responses to all three variants we checked, that is alpha, beta, and delta. However, the levels were quite significantly reduced compared to the wild type. This is the data for our T cell studies, and we saw again, as previously with the AstraZeneca vaccination, the vaccine Sinopharm produced potent T cell responses. However, the responses were significantly lower compared to the AstraZeneca vaccine when we compared the Sinopharm vaccine in this cohort. So, ladies and gentlemen, in summary, as we observed with Covishield vaccination, Sinopharm also produced robust antibody responses as well as T cell responses after 6 weeks of receiving vaccination. The third vaccine we introduced to our population was the Sputnik 5, and it was given to people in Kandy area. Again, as expected, Sinopharm Sputnik 5 also had a very good antibody response. However, the seroconversion rate was significantly lower compared to the other two platforms. And interestingly, with increasing age, the antibody index is significantly reduced, and there was a of the antibody response. The other vaccine.
Yes, somebody please mute the mic.
And we saw the using hemoglobination test, the titers for Sputnik 5 vaccines were similar to wild type, B117 and Delta, and these were comparable to the findings we observed with AstraZeneca vaccination. These are our data on our T cell studies, and we again saw high levels of S1 interferon gamma response, but we didn't see much of S2 response with regard to Sputnik 5 vaccination. So in summary, as with the other two vaccines, Sputnik 5 also produced significantly high levels of antibodies, and the T cell responses and the B cell responses were also robust. However, when compared to the AstraZeneca and the Sinopharm vaccination, the seroconversion rate, especially in the elderly population, was significantly lower. So we observed quite a lot of breakthrough cases, not only in Sri Lanka, throughout the world. And I have listed few possible scenarios for these breakout cases, probably due to the partial escape from the neutralizing antibodies and the high replication capacity, especially in the Delta variant, and the short incubation period. And sometimes, because of the viral load, the time, there might not be sufficient time for the memory cells to get activated and give the protective immunity to prevent an infection, but they will definitely provide protection against severe disease and death. So as with the vaccination, everyone thought we will have a smooth journey, but with the development of different variants, especially the variants of concerns, alpha, beta, delta, and gamma. So these are hindering our drive towards eliminating COVID-19 in the world. Especially, these are the key research questions that we need to answer. What are the absolute correlates of protection with regard to antibodies and also with regard to T and B cells, and the contribution of CD4 and CD cells in providing the absolute protection and the protective immunity from COVID-19. And especially in a country like ours, where we use multiple platforms, it's important to understand the difference in these mechanisms in giving protective immunity and the different platforms. Let me conclude with the current situation in Sri Lanka. We are the only sequencing facility for the country, and you can beautifully see how different variants, spread in Sri Lanka. We started with our own Sri Lankan variant, then we had a massive outbreak with the alpha, and then we had a massive peak. We are currently experiencing with Delta variant. These are a few more slides just to highlight the same fact. You can see here, Delta is dominating in our country, and you can see we have three peaks, especially with the Delta peak. You can see the deaths, number of deaths experiencing in our country is very high, especially in a situation where we had about more than 50% people vaccinated. And this lower part of the slide is the latest finding which I got from Nature today, and it says in vitro studies showed that Delta is 6-fold less sensitive to serum neutralizing antibodies from recovered individuals, which may be one of the reasons for the people to develop breakthrough infection, especially with the vaccination. It is 8-fold less sensitive to vaccine-induced antibodies when you are experiencing a Delta infection. So this is the latest report we issued to the Minister of Health today. You can see that our spread of Delta throughout the country, and you very rarely find a bit of alpha in the northern province, eastern province, and the central province and Sabaragamuwa. And apart from that, all the other areas of Sri Lanka, except where we didn't receive samples, are 95% are from Delta variant. So things are not looking good because of several reasons I have mentioned earlier in my slides. So I would like to conclude with that, acknowledging my team leader, Professor Malavigi, and our team members, and also our funding organizations, World Health Organization, UK Medical Research Council, Foreign and Commonwealth Office for Support, and NIH USA for unlimited funding to carry out this research. Thank you very much.
Yeah, thank you, Dr. Jeewandara, for that presentation. Now we'll have the next one.
Okay. Let me introduce the second speaker for today's sessions, that is Dr. Thushan de Silva. Dr. Thushan de Silva is a senior clinical lecturer in infectious diseases at the University of Sheffield, UK, and a consultant physician at the South Yorkshire Regional Department of Infection and Tropical Medicine. He obtained his primary medical degree from the University of Bristol, UK, and then trained in general internal medicine, infectious diseases, and HIV in London and Oxford prior to moving to Sheffield for higher specialist training in infectious diseases, medical microbiology, and virology. He was awarded several research fellowships to complete his PhD in HIV-2 molecular epidemiology and adaptive immunity at University College of London and the University of Oxford, which was the basis for his ongoing research interest in virology and viral immunology. Thushan was awarded the Royal College of Pathology Specialty Research Medal in Medical Microbiology and Virology for work during his PhD. Given his interest and expertise in respiratory viruses, in the last year, much of Thushan's time has been taken by SARS-CoV-2 research with viral sequencing and studies on T-cell immunology, along with active clinical service in COVID-19 wards in the NHS. He was awarded an MBE in the Queen's Birthday Honours List in 2021 for services to COVID-19 research. Thushan continues to be a visiting scientist and principal investigator in the vaccines and immunology team at the MRC, Gambia, and honorary associate professor at the London School of Hygiene and Tropical Medicine. Over to you, Thushan.
Thank you very much for the generous introduction, and thank you for the invitation to speak. Can you see my slides?
Yes, we can see.
Okay, perfect. Thank you very much. So I'm going to speak to you today about some of the viral sequencing that we've been involved with during the COVID-19 pandemic, and the previous talk has set this talk up very nicely. So thank you to Chandima for that. And I'm going to speak mostly about our experience in the UK with SARS-CoV-2 sequencing as one of the sort of early adopters of this technology platform for use during the pandemic, and speak about the multiple uses of viral sequencing that have really been pioneering in the context of a pandemic and has not really been done to this extent before. And I'll focus on two specific aspects where it's been used, so outbreak investigation and for public health surveillance, and hope to convince you that really not only has it played a key part in control and tracking of the pandemic to date, but really there's a need to upscale this capacity globally, and that it can really play a key part in future control. So just so everyone is clear what we're talking about here, viral sequencing essentially involves working out what the genetic code is of SARS-CoV-2. And now there are over 2 million genome sequences globally, and it has become the most sequenced pathogen in history. And we and many others have done this using something called the Arctic Network Protocol, where the genome of SARS-CoV-2 is shown on the bottom, where it's amplified using a technique of 98 overlapping amplicons to generate a complete sequence. And using this, not only can we read the sort of the DNA, RNA, and DNA code, but we can also look at the predicted protein structure during this, which is very important for thinking about phenotypic changes, as I'll come on to. So one thing that's really helped this effort has also been the technology that is available now. So so-called third generation sequencing technology using Oxford Nanopore technology, and shown on the right, on the top right is a gridiron, which is a device that's used in our lab. But actually there are many different versions, and you have palm top devices like the MinION on the bottom right, which can do the job just as well. And there are many benefits to this technology, but it's one of the reasons why so many labs in the world have been able to sequence SARS-CoV-2 to this extent. It's scalable, so unlike former technology, you don't have to wait for a lot of samples to come in before you do the sequencing, which means that you can run few samples and get a quicker response, quicker result. It's accessible to many smaller laboratories and by virtue to many countries which may not have had this technology before. And importantly, real-time analysis is possible. So our GridION is linked to our bioinformatic pipeline, and in many cases, we were calling variants that were in samples from hospital patients before the entire sequencing run was complete. So you can see how that would reduce the turnaround time and get a result more quickly. And the portability of this technology is demonstrated really by many uses of it in the field. So in the bottom left, I think it is sequencing done in the field during a Zika virus outbreak in South America. And even more extreme, on the picture in the middle is Dr. Kate Rubin's in the International Space Station, where a Mirani was taken to space, where the first sequencing was done using this technology. So our journey into this started in March 2020. In the middle there is the hospital I work in. On the 5th floor is the high consequence infectious disease unit. And we admitted 2 of the first patients in the UK at the time, which were imported infections. And while we were taking care of these patients, we realized that really we should be setting up the ability to sequence these viruses. And that's what we did, putting a group together across our university and the technology that was available. And we sequenced the first 2 genomes. And at that time, there were probably 30 or 40 patients in the whole of the country. And very soon we were part of the COG UK, COVID-19 Genomics UK consortium, and sequencing 100 to 200 a week, and have now sequenced over 12,000 samples from hospitalized patients, as well as some in the community and healthcare workers. And the COG UK consortium really, along with the Oxford vaccine trial and the recovery study, are probably one of the greatest sort of achievements in the UK during the pandemic. It really demonstrated how collaborative science has come together to try and tackle the pandemic. So COG UK was a consortium of 16 either academic institutions and public health organisations, as well as the Wellcome Sanger Institute, led by Professor Sharon Peacock. And the structure that was set up is shown in this infographic. So in the UK, most of the community testing is done in the so-called Lighthouse labs, and these samples were sequenced at the Wellcome Sanger Institute. And we were one of the regional sequencing sites where most of the hospital samples, as well as samples from healthcare workers, are sequenced. And these are brought together in a centralized cloud infrastructure and combined with electronic health records and information from the public health agencies, given to analysis teams, and then information fed back not only to the public health agencies, but ultimately to the UK government to make decisions on things like non-pharmaceutical interventions. But what was really key locally and utilizing the ability of some of the technology I described is that we were able to feed the information back directly to either local public health or the hospital infection control teams to try and utilize sequencing to actually make decisions that may impact what happened during the pandemic. And this summarizes a huge amount of work from lots of people. And the analogy that has been often used for putting this together is trying to build a plane while it's taking off already on the runway. Happy to say now that most of this is complete and it's functioning very well. So these are the 4 aspects that COG UK set out to achieve. And it always sat straddling 2 objectives: one, helping public health and decisions as the pandemic went on, and the other is research outputs. And often there was much overlap between them. And I'm just going to focus, take some examples from some of these objectives where we've been involved to try and demonstrate how viral sequencing has been useful. So firstly, in transmission and outbreak investigation, it's been used both by us and many others in both hospital and community outbreak investigations. And I'll give you an example of what I mean by this. So imagine there's a cluster of people with positive COVID tests, either in a hospital ward or a community. In the setting of wide circulation, it's very hard to know whether there's been a breach in transmission, in infection control, and the transmission has happened, or if these are all independent infections that have been acquired from many sources. So outbreak investigations may take place, which may include contact tracing and PCR testing. And this then feeds into rapid turnaround sequencing for us to then say whether all those infections form a genetic cluster or not, or if they're independent infections, and then decisions made on either public health interventions or in-hospital infection control. So how would we use sequencing to define genetic clusters? Well, as many of you will know, what happens is as pathogens and viruses in particular spread through individuals, they pick up mutations in their genetic code. Essentially, there are footprints in the history of transmission. And what we and others can do is use this information to link infections together. So here we can see that these first 3 infections share a lot of the same genetic code, and we can track that they've probably been either related infections, and in some cases the direction of infection from one person to the other. But we can see that the genetic code of the last patient is very different. So here we can say that these 3 individuals are probably linked, but not this individual. And unfortunately, in the UK and many other places, there's been a large burden of hospital-acquired COVID. It's estimated in some analyses that 1 in 10 of all COVID patients in hospital were actually acquired in hospital. And added to that, many NHS hospital staff acquired COVID. What we have less insight into is actually the routes of transmission. Where did these individuals get infection? How did that happen? And finding this information out can hopefully help learn from the past to put different measures in place for the future. So we decided to use the wealth of genomic information we had generated to really try and answer the question which hasn't been tackled much, which is who infected whom in large sort of outbreaks. And as many of you will know, what's difficult often is to decide whether infections are community or hospital acquired because of the long incubation period that SARS-CoV-2 has. Shown on the left here is essentially the epidemic curves during our hospital, along with on the bottom the evolving infection control measures that were put in place. So for example, fairly early on in the pandemic, there was obviously testing of symptomatic staff and cases, which soon evolved to testing everyone who came in through the front door. But then more latter interventions like lateral flow testing for staff and obviously vaccination later in the pandemic. And what we did was we combined the genomic and epidemiological hospital epidemiology and location information from over 2,000 patients and staff in a Bayesian mathematical model in conjunction with colleagues at the London School of Hygiene and Tropical Medicine to really see where these infections have been acquired from. And what we found was that although there were some patient to staff infections during the first wave in the UK, most staff acquired their infection from other staff. And this transmission type was the commonest in that wave, but that reduced dramatically during wave 2, potentially as measures like social distancing in hospitals and universal mask wearing in non-clinical areas came into fruition. But unfortunately, during the second wave, patient-to-patient infections were incredibly common, and they went up dramatically from the first wave. And just to summarize, what we then went on to see is where patients were acquiring their infections from. So what we found is that actually the onward transmission from community-acquired cases coming into hospital, so already with the label of SARS-CoV-2, was actually very minimal. It was only 4% of those cases would then transmit on to someone else, whether it's staff or patients. But the hospital-acquired cases were actually then over about half of them were transmitting on. And this was despite the fact that all individuals are being tested at admission and routinely at day 5. So this really showed us that despite these measures, that hospital-acquired infections were slipping through the net, and by the time they were discovered, it was already too late and they had transmitted on. So because of this, various measures have been put in place, like rapid antigen testing and more frequent testing of individuals to try and prevent this now during the third wave. And this is work from Ben Lindsay, one of our clinical fellows, along with a lot of other individuals. So moving on to genomic surveillance and mutational analysis and tracking, and I'm going to tackle both of these two things at once because they're related. Many of you, and as Chandima alluded to, now we all know that there are new variants of SARS-CoV-2. For example, the Alpha variant first detected in the UK, the Beta variant first detected in South Africa, and more recently the Delta variant. So what do we mean when we say variants of concern, and why are they important? Because there are many reasons why they may be. So as many of you will know, most mutations that SARS-CoV-2 acquires do not have an effect on the behavior of the virus. So we're talking here about mutations that do have an impact either on increased transmissibility, increased disease severity, or perhaps most importantly, the reduced susceptibility to either natural or vaccine-acquired immunity. And determining through thorough sequencing of what is happening can have impacts on your decisions about non-pharmaceutical interventions to limit the impact on healthcare systems. So, for example, in the UK, when it was discovered that most of the infection rise during the latter part of 2020 was due to a new variant Alpha that appeared to be more transmissible, there were decisions taken about extending restrictions for longer. And moving forward, the real relevance here of genomic surveillance and thorough genomic surveillance is really going to be assessing the threat to vaccine programs as we move forward. So again, through sequencing, we've been able to really track like never before. And you know, just to highlight, this is the first time in history that we've been able to track the evolution of a pathogen from its emergence. Usually we're looking back in history to see what happened. And in this gray area, essentially a long period of time from the start of the pandemic where there were really not many changes that affected virus behavior. And the only one really that did is this D614G mutation, which appeared to have some increased ability to transmit. And very early on, we were involved in some work with individuals and colleagues in Los Alamos. So Betty Korber, they contacted us because we had deposited quite a lot of the early sequences from the UK to say that this variant or this mutation appeared to be taking over. So there was a shift wherever it landed in the world. Soon the D614 variant disappeared and G614 appeared. And we were able to see in swabs from our local hospital that there were higher virus levels in individuals who had the G614 mutation. But importantly, these infections were not overrepresented in our hospitalized or severe cases. And in even better news, again, we were able to provide some serum and patients for this study, again, with colleagues in the US. Actually, it seemed that this mutation resulted in the virus being more susceptible to antibody neutralization than less. So it seemed that there was some early adaptation of the virus, which we and others were able to track. And then for many months, there were really no changes that had any impact on the biology of the virus. But from around sort of October, November 2020, and we can only hypothesize that this may be due to the fact, partly due to the fact that there's increasing population immunity, there was increasing selective pressure and variants of concern started to appear. So Alpha, as you've heard, first described in the UK, was the major one to then shift the sort of nature of the pandemic. And again, this took over from previous lineages wherever it appeared. And we saw that there was increased transmissibility. There's good evidence for increased severity, unfortunately, with this variant. And there are a number of reasons why this might be. But fortunately, there was really not a great impact on antibody recognition, certainly not enough to threaten vaccine programs. But more recently, as you've heard, now the Delta variant has appeared, and there's good evidence for much greater transmissibility than the Alpha variant, and the basic reproductive number, so the number of onward transmission from one case, is estimated to be around 7 compared to about 3 of the former variants. There's some evidence for increased severity, but that hasn't been replicated and not convincing yet. But importantly, there's now probably the first sort of widespread variant that has some impact on antibody recognition. And the former speaker has already shown this, and we've worked with him and Professor Malavige and been fortunate to do that to try and describe what's been happening in Sri Lanka. And I've put this up mainly to show that the B1411 variant, the lineage, is one that really caused the bulk of infections and spread throughout the country in autumn, so September, October last year. But this really didn't have any sort of biological impact. There was no phenotypic change. So the reason it spread most widely initially was probably because of what's called founder effect. So this was a virus that was around at the time that then spread widely. But since then, Alpha has taken over, and as you see with more up-to-date data, Delta has taken over. And just to orient you to some of the similar data to the former talk, what we've been involved in a large multi-center cohort study in healthcare workers, really tracking what's been happening to Pfizer and AstraZeneca vaccine responses in individuals. And as you may know, even for the Pfizer vaccine, the UK took a decision fairly early on to extend the duration from the licensed course of 2 vaccines 3 to 4 weeks apart to 10 to 12 weeks apart. And what we've done here as part of one of our studies is to look to see what happens after the first dose, what happens then in that interval between the first and second dose, and what happens after the second dose of the vaccine. And here's Victoria as a very early Wuhan-like lineage, and then the beta, gamma, and delta. And importantly, with regards to my talk, is really you can see that after 1 dose, and certainly during the long interval, there's really no neutralizing activity detectable to the beta variant. This is using live virus neutralization assays, whereas to gamma and Victoria, there's reasonable levels. And with delta, there's a reduced antibody response and neutralization after 1 dose, which then disappears completely and wanes during this long interval. But thankfully, after 2 doses, there's a reasonable response. So this is really what our focus as a sort of global scientific community in terms of this aspect of pandemic control will be. And we know quite a lot now about the spike or the surface sort of protein of SARS-CoV-2 shown in different variants on the left, and what some of these mutations may be. And what we're seeing are now recurrent appearance of mutations because sequencing is done so thoroughly. So for example, this N501Y mutation that has appeared in several variants of concern, which we know now increases affinity for host cell binding. There is increased efficiency of furin cleavage with the P681H mutation in alpha, and delta isn't on there, but this is one of the hypotheses why delta may be far more transmissible. And now increasing mutations where there's reduced neutralization by sera or monoclonal antibodies. And what we're seeing now is what's called convergent evolution. So the appearance of these mutations in different parts of the family tree of SARS-CoV-2. So these mutations are appearing independently, not because they're originally related. So what is next? And that's really the question moving forward, which we don't know the answer to, but we can get some idea from what might happen from other seasonal coronaviruses. So shown on the left here is basically a family or phylogenetic tree of the alpha coronavirus 229E, which has been identified in humans over 50 years ago. And this ladder-like appearance of the family tree basically shows what happened, where a sequential evolutionary variant appears probably in response to antibody pressure over time. And if you take sera from 1984, that sera cannot neutralize current day viruses for the alpha virus as well as the viruses from 1984, showing sequential immune escape. So this potentially is what might happen, and this is what we need to really keep a close eye on moving forward. And now we know so much about, quite a lot about what individual amino acid mutations might do in spike, surveillance is really essential, looking for emergence of these constellations of mutations that might define new variants of concern. So to end with, really, just to make a plea, I think, you know, it's been very useful in the pandemic so far, but really the sequencing capacity globally needs improving urgently. And this is taken from an editorial that Nilika and I wrote for the Sri Lankan Journal of Medicine recently, showing that most of the sequencing has come from high-income countries to date, whether you look at the total number of sequences or the percent reported cases sequenced. Sri Lanka has been doing very well, and I'm sure this number has gone up since June in South Asia. But really, you know, we need to improve this globally to really be able to track variants as they emerge, if they emerge in different countries. And really, this data flow and information needs to be done in a way that it can aid public health decision-making in real time. And this has a relevance not only for national vaccine programs, but it has a relevance for global pandemic control. Because if we don't have sequencing coverage all over the world, there's going to be sequencing blind spots, and we're going to detect variants of concern only when they spill over into countries where there's high sequencing coverage, by which time it may be too late in the country of origin, and it may threaten vaccine programs. And this needs to be done. Lots of things need to happen for this to be achieved globally: improve laboratory and bioinformatic capacity, but also data flow done in a way where it can be fed back to relevant stakeholders immediately. And to help this, the WHO came out with a very, very thorough and useful guide to genomic sequencing and scaling up and establishment across the world. So finally, pathogen genomics and SARS-CoV-2 sequencing has been very good for us to learn from the past, like the hospital example that I gave you. But really, it has been used for the first time to try and understand the present. And this is what we really need to do and aid most countries to be able to do this. But clearly, what we also need to use it for, if we can, is to try and predict the future. And so we can try and be one step ahead of the virus as we move into the next phase. So I just want to finish with thanking our group in Sheffield, as well as Nilika's group for our collaborative work, and the mathematical models from the London School of Hygiene and Tropical Medicine. Thank you very much.
Thank you, Thushan, for that clear presentation. We'll get on with the next now.
Let me introduce the third speaker. I'm happy to introduce her as one of my colleagues in KDU. Professor Rohini Fernandopulle is the Senior Professor of Clinical Pharmacology and Therapeutics at General Sir John Kotalawala Defence University, and Emeritus Professor, University of Colombo, and WHO National Consultant on Regulatory Functions to NMRA, Sri Lanka. She obtained her MBBS from University of Ceylon and obtained her PhD in Pharmacology from University of Colombo, an honorary fellowship of the College of General Practitioners, Sri Lanka. She was the professor in clinical pharmacology and therapeutics of the Faculty of Medicine, University of Colombo, and also served as the professor of pharmacology at the International Medical University, Kuala Lumpur, Malaysia in 2007, prior to her current appointment. She has more than 30 journal articles in peer-reviewed index journals and has contributed to more than 10 WHO publications, 20 booklets, more than 80 conference papers and abstracts, including 11 award-winning abstracts. She is currently chairperson of Ministerial Committee for formulating the Essential Drug List Sri Lanka and the secretary of many committees at NMRA and member of many national advisory committees. She holds the position of World Health Organization National Consultant, Monitoring and Evaluation of the Regulatory Functions of NMRA from 2020, and had been a member and an advisor and a resource person of many expert committees of WHO and other international organizations. Over to you, Rohini.
Thank you, Mirani, for that generous introduction. And also thank you to the organizing committee of the IRC, chair in the committee, for having given me the opportunity to present the lessons learned by dissecting the challenges posed by COVID-19 to pharmaceutical regulation in Sri Lanka. Since this is the presentation on pharmaceuticals, I declare no conflicts of interest, and I also state that these are my views and not the views of the WHO or the NMRA. These are presentation outline. I will first introduce you into the current regulatory pathways in the country. So the normal registration pathway, we have 3 pathways at present, which is where we get a dossier, we do a full evaluation, and the condition to register a product in Sri Lanka is that it should be approved by one of the regulatory authorities recognized by the NMRA. And following that, you do a full evaluation of both the pharmaceutical, non-clinical, and the clinical evaluation, and that usually takes about 300 working days. Then we have 2 other mechanisms which are for priority. This is the reliance pathway that is given by WHO. For that pathway, we have the full dossier. It has to be approved by one reference agency, and that agency has to give us their evaluation report. We look at the rest of it and determine the quality, safety, and need for our country, and that takes around 120 working days. This is the last one, which is a verification process, because it is approved by 2 reference authorities. We really don't go into detail of that, but we go through the reports and see that the company has submitted all the data to the NMRA, and if we are satisfied with the data that has been submitted, it is given either rejected or given registration, but it takes 90 working days. Okay, so these pathways are not really enabling us to give recognition for these vaccines and medicines quickly. So therefore, we looked at the National Medicines Regulatory Act of 2015, and clause 109 gave us the conditions to grant permission in special circumstances, such as to save a life, to control an outbreak of an infection or an epidemic or any other national emergency of a national security, to import and supply a particular medicine, medical device, or borderline in specified quantities. To use this clause, the request had to be made by the Ministry of Health or by any individual and organization recommended by the Ministry of Health. So this was the clause we used to expedite registration during the pandemic. So at the same time, though we were trying to expedite the regulation and make products available, we did not want to compromise safety, efficacy, and quality standards, and also the need for our country, because we are resource limited. Some of these are very expensive medicines. So therefore, we look for the clause in regulation that was could have been used for this COVID pandemic, and then that was sent to the Ministry of Health for gazetting. So the NMRA then also decided on how we are going to utilize this clause, and therefore without compromising safety, efficacy, quality standards, and also the need for our country. So therefore we decided on different pathways. One is the reliance pathway, which is the one that has been approved by the World Health Organization. And we appointed eminent researchers, academic consultants in advisory committees for evaluation of devices, vaccines, and medicines submitted for the emergency use permission, and of course shortened all regulatory timelines. So this is the list of WHO. The WHO for this particular pandemic, the WHO listed authorities for reliance on COVID-19 vaccines. And as you can see, it's either WHO emergency use license or pre-qualification, and others were the leading stringent authorities like Australia, Belgium, Canada, EU, France, Germany, Italy, Netherlands, Switzerland, UK, and USA. So now I will go through what the challenges we faced with vaccines. Now, I'm quoting from John Cohen from the Science magazine. He says vaccine development, manufacture, and deliverance under 1 year is a notable scientific feat in year 2020. So why did he say that? Because normally, traditionally, the current vaccines we use now, it takes 15 years or longer for it to reach get regulatory authority for them to be used in the country. Here, the SARS vaccine was developed. The total time for development was 10 months to 1.5 years. So it's a real feat. And it's also a feat for a resource-limited NMRA with very few people there to have been able to expedite all these evaluations and grant emergency use permission. To help national regulatory authorities to do this, the Strategic Advisory Group experts SAGE on immunization, this is a group from the WHO, they released on the 10th of December the framework to inform assessment of evidence and formulation of subsequent COVID-19 vaccine recommendations. So this was based on the PICO concept. All of you are aware of PICO, where it is the patient or population, I for intervention, C for comparison, and O for outcome. So the comparison was with placebos. And should the COVID vaccine be used in adults greater than 18 to 59 years? Should COVID vaccine be used in older adults greater than 60 years? And should COVID vaccine be used with individuals with comorbidities or health states that increase risk for severe COVID-19? And the WHO was the standard they adapted was 50% efficacy, of course, preferably closer to about 70% with no serious harms. So at the beginning of the pandemic, Sri Lanka did very well, as was told by all the speakers. And therefore, we were not really, at the beginning, we were not pushing hard for vaccines. It was only later on that when the thing was accelerating that we started looking for vaccines. And at that time, not that we were very much later, but if you can see, it's around 10th of October, in October 2020, that these vaccines were registered for use in the stringent authorities like US FDA and the European Medicines Union and also in UK. So this was the Pfizer vaccine. The efficacy was over 93% in all the age groups. And the vaccine area is the AstraZeneca vaccine, which was developed by Oxford and AstraZeneca, and it had an 83.8% overall. So that was the data we had when we got the expression of interest for vaccines in early January 2021. So the first vaccine that came for registration to Sri Lanka was the one that was developed by Oxford and AstraZeneca, but it was named Covishield because AstraZeneca transferred the technology to Serum Institute of India, and the name was Covishield. And so Serum Institute of India at the same time submitted the dossier to Sri Lanka and to the WHO also for their approval. So we got we gave the approval on the 22nd of January. So you can see how quickly we did it. And the European, the emergency was given after us on the 10th of February. So I told you we appointed a committee of experts, and they really worked very hard day and night. And these were the key points in the evaluation report of the National Medicines Regulatory Authority in Sri Lanka, which said that the phase 1, 2, 3 clinical trials were published in peer-reviewed journals. So then overall efficacy was 62%, but different studies gave different rates, and it went up to about 80%. then we also detected that if the gap was widened, that the efficacy was better, so over 6 weeks. And we also got interim data from ongoing bridging phase 3 studies in India with 1,600 participants. That was done by Serum Institute. And they also looked out, if you go back to the questions, they looked for the over 65 age group and also for those with comorbidity, and found that efficacy for comorbidity was similar to the general population, and that the number of participants over 65, though small, showed that immune response was well documented. And the animal studies, what the committee was very worried about was vaccine-enhanced disease. They had shown enough animal data to show that this was not a problem with this vaccine. Now, if we look at the EU, the WHO emergency utilization authorization, which was given after us, same points are reflected. It said that it is effective. It said that it tend to be higher when the interval was longer, that those small in the over 65-year age group, it was effective. Those with pre-existing comorbidity had similar efficacy. And they also said that no precise efficacy estimates for this age group are expected soon. So there wasn't very much different from our one, and we were very confident that we had given a good good report and approved, given an emergency use permission on a valid report, which was similar to the WHO, which we did before WHO. Then came Sinopharm COVID-19 vaccine. It was produced by Beijing Bioinstitute of Biological Products, subsidiary of China. It was a different vaccine. It was inactivated vaccine, Weerasooriya vaccine, and it was authorized in China on the 31st of December, and we got the expression of interest on the 6th of February. And one of the big challenges the NMRA faced was language barrier, because we could not communicate effectively. And also we were not sure whether it was the same dossier that was submitted to the WHO, because there was some data that was lacking. And even the phase 3 data was not published in a peer-reviewed journal. So the key points in the NMRA report was that the phase 1 and 2 have been submitted, which was okay, and we agreed on that. But the phase 3 data was only an interim phase and it was not complete. And the main problem that we found was efficacy data in over 60-year-old age group. The sample size was very small and it was not adequate to draw conclusions. And even in the comorbidities, it was a bit difficult, because from what we understood from the dossier that had been submitted, it looked like they had been excluded from the study. So detailed information on the adverse events also was not very clearly specified. And even the risk of possible vaccine-associated enhanced disease was not addressed to the extent that we would like to have had. So if you look at, but WHO took long time on this, because they did, I think, they went personally to the manufacturing site and did a WHO GMP, and it was after that that they looked at the clinical data. So the EU was granted on the 7th of May. So as a result, there was a worry that, the NMRA was delaying registering the Sinopharm vaccine. But it was really on genuine grounds that it was taking time. And so that report says they are also very confident that 2 doses of the vaccine is efficacious in 18 to 59. They are only moderately confident that the risk of serious adverse events following 1 or 2 doses is low. And that low confidence in the quality of evidence that 2 doses are efficacious in preventing PCR confirmed COVID-19 in older adults, and very low confidence in that the risk of serious adverse events following 1 or 2 doses in adults, in the older adults over 60 years. But WHO, with their experience and their knowledge, and they are looking at the global situation, they gave the emergency use authorization, but they gave this with a clause which said that recommends that countries using the vaccine in older age groups should conduct safety and effectiveness. That is, on the effectiveness is on the ground situation when it is being given, to make recommendations more robust. So therefore, that's how the emergency use authorization was given by the WHO. So learning from that, I think Sri Lanka being a small be a regulatory authority, we have never had this experience before. We were very cautious. Maybe we were too cautious, and we therefore kept on asking for more data. Whereas the WHO taught us a lesson that in an emergency pandemic situation, you had to look at benefit at large. So it had given over 50% efficacy, but maybe we could have looked at other things by telling them to have stringent post-marketing surveillance strengthening and reporting it to the authority. So also this could have been a consultative discussion with policy makers, because later on it became very confrontational, which should be avoided in the future. So herbal medicine, I'm not going to talk too much about herbal medicine because our Act explicitly excludes Ayurveda and homeopathic medicines. But I think that for clinical trials, we should try to get them in and have a collaboration between allopathic health professionals and Ayurveda and traditional medical practitioners is the way forward, because to guide them on good clinical practice guidelines without authorizing unproven products to be used in the community in a pandemic situation. So finally, I'm dissecting the challenges posed by COVID-19 pharmaceutical regulation medicines in Sri Lanka. So I'm focusing on ivermectin. When the pandemic was accelerating, there was unofficial information that the NMRA got that ivermectin was being used and it had come into the country without registration, and that people were paying exorbitant prices. The one that I was able to find was around 250 and 1,000 rupee within that range, but I'm sure it went even beyond that in certain outstation places. And these are baggage goods that we did not know whether they were really genuine products or even spurious products. Then we looked at WHO recommendation. WHO recommends only to be used within clinical trials. And there are a lot of ongoing trials globally assessing ivermectin. WHO specifically states that it does not look at prevention because it's out of the scope of the current WHO treatment guidelines. And a lot of the stringent authorities, of course, also did not recommend because of the fact that there was no solid clinical trial data. But on the other hand, there were health professionals in a lot of countries, including the stringent authorities, which were giving a different opinion. So most of these countries, like in Sri Lanka, had divided opinion. And then we also had a letter written by a very senior academic, a physician, on its use in specified groups. So when as a National Medicine Regulatory Authority, we had to look what we should do. And when we looked at the literature what was available around the beginning of August, there were several reports which the British Ivermectin Recommendation Development Panel recommends for the prevention and treatment of COVID-19 to reduce morbidity and mortality associated with COVID-19. And in fact, in this meta-analysis, they had used a study which was done in Brazil, which was later removed, but they made a public statement that in spite of that to show that it can be used. Then All India Medical Institute published a mass case study in +1, where they said that 2 doses of ivermectin prophylaxis at a dose of 300 micrograms per kilogram, that was weight-based, with a gap of 72 hours was associated with a 73% reduction among healthcare workers for the following month. And this was published in a peer-reviewed journal in PLOS. Then we had recently another one that came from the American Journal of Therapeutics. It said that moderate certainty evidence finds that large reductions of COVID-19 deaths are possible using ivermectin. And finally, we had this from the New Microbe New Infection Journal, where Professor Boradi, who is a very world famous professor, who really came out with the triple therapy for peptic ulcer disease, who also said it's a multifaceted drug of Nobel Prize distinction. So therefore, we were in a dilemma whether we should allow this to come the proper way into the country or ban it. Banning is it's not easy, because in spite of all the regulations, it's still coming in baggage. We know several medicines coming to this country without registration, even though this is not allowed legally. So then, are we going to allow patients to swallow veterinary? Ivermectin is used a lot in the veterinary sector. So we didn't know whether it was a veterinary product that was coming in or human product. Then at that time, there were applications for waiver of registration from market authorization holders who bring medicines to the country and who have not had quality failures and issues in that way. So we, as an NMRA, thought we should allow this, should do something about it. Either we have to stop it or allow it to come and make sure that whatever is available for the public is quality assured. So therefore, the NMRA decision was, we looked at the Sri Lanka National COVID-19 treatment guidelines. It's not recommended in our guideline. So we decided to grant waiver of registration, not emergency use permission. We didn't give emergency use permission. We only granted a waiver of registration by looking at some critical quality aspects, because obviously all the whole dossier didn't come. So we looked at the important critical quality aspects, and then we decided to give the waiver of registration. We negotiated prices, and you can see prices of ivermectin drastically came down, and now it is available around 32 rupees, ranging from 32 to about 50. I think recently it was even 25. Now we are having similar problems with other medicines. Those are recommended and not recommended. Now, tocilizumab is recommended for severe critical COVID-19. It is registered. It is registered for another indication, but we can allow it for an off-label indication. But the issue was that the market authorization holder has only limited stocks, and he couldn't supply what the country needed. So then again, it was coming through baggage. We didn't know how they were storing. These are parenteral preparations. So we, as an NMRA, we had to do something very proactive. Then remdesivir, it was first not approved, and now it has been approved in EMA, US FDA, and several other countries. Then we also have casirivimab and imdimimab, which is a combination approved in a lot of regulatory authorities. It's very expensive. All these drugs are expensive. They are not cheap. And this is for prevention. And we also have the College of Pulmonologists, who was asking us to register this combination for people with transplants who needed the medicines to prevent the disease. So, and also we knew that it was available in the private sector at exorbitant prices without either waiver of registration or emergency use permission. So then we looked at what ways we can do this, and now the WHO has given a mechanism following their experience with Ebola. When they had Ebola, it was similar. They didn't have treatments. So what they said was, the guideline says for monitored emergency use of unregistered medicine and investigational interventions, which is for MURI, for COVID, it is for Ebola. I have changed it for COVID-19 virus disease, which is going to be implemented by NMRA. So we have, there's an expert committee looking at this, and we have just finished casirivimab and imdimimab, and we are writing a lot of, you know, what the market authorization has to do, how it should be monitored, what the doctor should do, each person who's given it should be documented. So we are working out a legal mechanism, and we will give the waiver of registration or emergency use, very likely emergency use permission, because if they submit the full dossier, I think they have submitted. So what are the lessons we have learned? One lesson is that Sri Lanka has eminent physicians, scientists, and physicians to evaluate safety, efficacy, and quality of our medicines, especially in pandemic situations. And now from this experience, I'm sure all of the people who have taken part in these exercises will be able to make proactive decisions which are beneficial for the country. And these should be in consultation with the policymakers. And we should, NMRA did not have, when this pandemic unfolded, we did not have a proactive policy for this sort of situation. So therefore, we probably did not start securing strategic stocks of medicines, vaccines, medical devices early by engaging with policymakers and the industry, because they are the ones who have to bring it in. Now we don't have method of forecasting requirements, because some of these are coming through baggage and we don't know how much is being used in the private sector. We have the ability to check what is in the state sector, but not in the private sector. Then even with ivermectin, we should have planned the clinical trial early, because there was need for this quite early. And now there is a trial going on, but now the peak is sort of now on the downward trend. So we should have planned this quite early after the Indian data was coming out. And closely track all potential vaccines, at least now from now onwards, we should closely track all potential vaccines and medicines in development together with the market authorization holders, so that we secure our stock of whatever vaccines, medicines needed for the country. The other issue from this, what we should learn is that the NMRA is acting quite alone. The healthcare professionals.
Yes, sir.
What happened?
Yeah, disturbance.
And here you, Rohini.
We should have a good mechanism to prevent this and build strong links, and finally proactively monitor unregistered pharmaceuticals in the market and misleading advertisements to prevent misuse of medicines in a pandemic situation. So that is all I have to tell you. So I acknowledge all the advisory committees of the NMRA who helped us in this situation. The chairman of NMRA, Dr. Rasita Vijayawanta, and Dr. Kamal Jai Singh, and particularly the staff of the NMRA, though they were not visible, they were working their time, they were contributing timeless service to the people of the country, risking their own lives to register, give this registration to all the medical devices and to the vaccine. And the former chairman of the NMRA, because this pandemic started during his time, for giving the leadership during the pandemic. Professor Thushan de Silva. Thank you.
Yes, I am privileged to introduce the last speaker. That is my teacher at University of Peradeniya, Dr. Palitha Abeykoon. Dr. Palitha Abeykoon currently serves as the WHO Director General Special Envoy for COVID-19 to Southeast Asia and is a senior advisor to the World Health Organization. He received a master's in medical education and a master's in education from the University of Southern California. He was also a Taro Takemi Senior Fellow of the Harvard School of Public Health in Boston, where he did the master's in public health. He was Director of Health Systems Development and Research in WHO Regional Office in New Delhi, and concurrently Dr. Abeykoon served as the WHO representative to India. A former president of the Sri Lanka Medical Association, is a fellow of the College of General Practitioners, the College of Community Physicians, the College of Medical Education, and also the College of Medical Administrators of Sri Lanka. Over to you, Dr. Palitha Abeykoon.
Good afternoon. Thank you very much for your very kind words. Co-chairpersons, Dean of the Medical Faculty, members of the academia, distinguished invitees, students, ladies and gentlemen. I'm extremely thankful to the Dean and the organizers of this seminar for the very kind invitation to speak on this very important topic. I have very pleasant memories of being invited on 2 earlier occasions to similar seminars, and I always recall how elegantly organized and well conducted sessions at the KDU are. Today, I plan to share a few reflections on the current COVID-19 pandemic and how I felt it has been managed. Since I am the last speaker, it is possible that I might be repeating some of the points that were made by the distinguished experts who preceded me. I do beg your pardon for that. Today, my presentation will outline a few aspects of the COVID-19 pandemic. I'll provide a quick overview of a number of aspects of the pandemic. I'll touch on its evolution, briefly the global and the local situations, refer to the public health strategies adopted during the phases, and provide a few suggestions as to how we could come out of this pandemic tunnel with a stronger public health system. This is a quick snapshot of the global situation. This is as of last Saturday when we prepared this. Up to then, 220 million confirmed cases were found in the world, at least a third more, I guess, with over 4.5 million deaths. On the other side, over 500 million doses of vaccine have been administered. Of course, with regard to vaccines, as we shall see later, there has been tremendous inequity. There are countries which are now giving the 3rd dose, thinking of a 3rd dose, some are giving it, and there are some countries in Africa who have not yet received a single dose. We'll see how Sri Lanka has done later. This is a quick snapshot of the Sri Lankan situation that all of you know very well. We have had, so far we have had about 458,000 or plus diagnosed cases. Many have recovered, of course. The figure of deaths here is as of 2 days ago. Now I think it's exceeded 10,000 in the active cases, and we have done over 5 million tests. These are the daily reported cases and deaths from January 1st, 2020 until now. I'll move over to the next slide, which gives a better view of this. We can see the so-called first wave, second wave, and the continuing third wave, as it says. The first case, as you see here, was reported in Sri Lanka on 27 January. And the first intrinsic case was on 11th March of the same year. And now we are in the end of the 3rd or 4th wave. During the early part of the pandemic, the country's strong public health system, with the support of the other agencies, was able to rapidly identify the cases, do the contact tracing and quarantine contracts, and provide care for everyone who were turned positive. Now, if we look at the progress of this shows the epi curve from the beginning till about the end of September. This is the end of the first wave, as we call it. And we see the 3 clusters that came up during that time, the Navy camp, the Kuwait flight cluster, as it was called at that time, and the Kandahar cluster. Now, during this time, as I mentioned earlier, the surveillance of COVID-19 during the first wave was extremely efficient, extremely good. And here we had the confirmation of cases by the designated laboratory for testing COVID. There were 9, I think there were government laboratories as well as private laboratories. And the epidemiology unit naturally was a national focal point for COVID-19 surveillance. They worked very closely with the community and the National Intelligence Service and the quarantine centers. The three units worked very harmoniously, and there was almost a seamless partnership between them. The community, which included the epidemiologists, the medical officer of health, the PHIs, and the field staff, who did the location, informed patients, arranged for admission, contact tracing, etc. The intelligence service facilitated the contact tracing and did human service during that time. And the quarantine centers, the medical staff of the armed forces were at the quarantine centers in the early stages. And they also, I think, as far as we know, did an excellent job. When we look at the three phases, we see the three waves, as we call them. The first wave, as I mentioned earlier, from March till October, we had 3,396 cases and 13 deaths during that 9 months. And we had island-wide lockdowns from 20th March to 26th March. This is very interesting, as we said, we should see later. And there were nighttime restrictions till about 28 June. Second wave, which started on the 4th of October and continued till the 15th of April. Here we had isolated lockdowns more in high-risk areas only. And during this period, in contrast, we had 92,000 plus cases and nearly 600 deaths. And of course, now we are going into the third and the continuation of the third wave. We will talk about this a little bit later. And we have here the new variable which has come, that is the Delta variant, which is, as we can see later, has practically overtaken the other earlier viruses that were circulating in the country. And this is, I'm sharing this slide to show you that in the first wave particularly, looking at the epi curve, Sri Lanka did extremely well. In fact, compared to all these countries, Australia, New Zealand, New Zealand is held as a model, but in terms of the first wave, I think Sri Lanka did much better than that in all the parameters that all the indicators that we take to measure. So that's the situation it was. Sri Lanka, in fact, I think was a top-ranked country for the response and its containment. Then we see the progress and the Delta variant taking over, the possibility of a sudden surge, and hints at the possibility of a sudden surge with other potential variants of concern, and the need to have a mechanism in place to rapidly detect and respond to such strong surveillance system with integrated real-time data became very apparent. And this is going to be my theme, why we need, in light of all this that we experience, why Sri Lanka needs to think of building back better and come out of the COVID tunnel stronger than when we entered it. This is just to show the number of PCR tests conducted and the test positivity rates. It follows the normal pattern that we have seen in other countries. And now I think our test positivity rate is around 25%. I think it's 26% here, been going up progressively. Of course, it confirms and correlates with the number of tests that were done at the same time. This is with regard to one of the basic things that we are interested in pandemics is the health system, the capacity of the health system to handle the cases that come in, and also to make sure that it is not overburdened. This is the figure that I found for the 12th of August. I want to just highlight one point here. This situation as of 12th of August, last one, we find that with regard to particularly the dedicated ICUs, there's nothing vacant. The other ICUs, which are not dedicated but available in the country, at that time there was one bed vacant. And similarly with regard to level 1, level 2, level 3, they were getting filled up pretty fast by that time. Now, what I want to show you here is that just 10 days before that, we had capacity. Dedicated ICUs, we had 14 vacant, occupied only 83 when we had a capacity of 98. Unlike on the 12th when nothing was available. This time we had only 89% occupation and 95. You know, what I want to say is that during these two slides show how the health system was getting quickly overwhelmed during the peak of the pandemic with the Delta wave, when all level 3 and ICUs are fully occupied, and how it had further worsened during just 1 week. And this is to show the situation at 2 points in time. This was 27th of June. I don't want to, it's a busy slide, I don't want to take too much time on this slide, but I want you to see the number of red marks or red blocks in this. And the overall, this was the time vaccination has already started. The overall comment is that we were at what you might call level 3, situation level 3 with regard to the burden on the health system. And there was high level of transmission with a decreasing trend. The risk of health system getting overwhelmed, risk was there, and public health and social measures needed to be reinforced. But the situation level was rated as 3. In contrast, we come to 22nd last month, which is about 3 weeks later. You can see all 3 segments: the level of transmission, assessment of the health system capacity, and assessment of vaccination, all are growing red now. And the overall assessment also is at level 4 now, situation level 4, which requires extensive measures to prevent the collapse of the health system, and which means the outbreak is more severe and the threat has increased. And the overall level of transmission is in the red category, health system, high risk of being overwhelmed is red category, and we had to continue with the public health and social measures along with the vaccination. This is to share with you what is referred to as Oxford Government Stringency Index. This has been constructed based on 9 sub-indices that you see on the side. These sub-indices have been further subdivided under 2 levels for the region and for the entire country. And what you see on the right are the different segments, the codes which are, and this just to show you how it is calculated and how it is how it is applied in the case of Sri Lanka. The Oxford COVID-19 government response tracker collects systematic information on policy measures that governments have taken to tackle COVID-19. The different policy responses are tracked since January 2020, and they cover more than 180 countries. And they take into account school closures, travel restrictions, vaccination policy, and these are recorded on a scale to reflect the extent of government action, and scores are aggregated into a suite of policy indices. And this data is generally used by policymakers to understand what the government responses should be. This is the Sri Lankan government stringency, how it was used and how it affected mobility and the daily reported cases. What would be interesting to see here for you, for all of us, is that when the stringency is increased, mobility decreases, and vice versa. It's quite clearly seen. And we need to highlight in the future, even if it clearly shows that with increased stringency, what we say lockdown, the word that is used commonly is lockdown measures, the mobility as seen here is a 7-day average mobility goes down, and so does the number of cases. This is seen very clearly, how the cases go down and how the cases go up once the mobility or the stringency level is relaxed. I think this is important in the current context when we are discussing what should be done each week. This is the application of the stringency index during the first, second, and third waves. Clearly, there is a relationship between the stringency level. See how we contain the cases very tightly in the first wave. Second wave, we relaxed halfway, and then in April, the thing blew up again. And then when the stringency level is increased, the cases go down, and it was relaxed, I think, end of July, I believe, and the third wave, the second part started. This is a model to indicate how this will change if the same stringency is maintained till 18th September. What we are having last week, if we maintain till the 18th of September, it projects that the number of cases from current amount will decline, having the highest numbers within the high stringency period, provided that a decent level of stringency is maintained up even after the 18th of September. What this shows is that if we maintain this level of stringency till the 18th, the number of cases and correspondingly the number of deaths will be declining by a large amount, And this is the stringency level that you see the yellow line here. If we relax it gradually, by about the 1st of December, we should have some very positive results. Otherwise, the danger of the cases rising again lurks in the background. This is to say, if we continue till maybe 1st of October or so, what is happening now, the cases will come down even more drastically. By about mid-November, we will be able to contain this to a large extent. And certainly Sri Lanka will move into what is called the, if not the green, the amber category by the end of October, by early November. This is something that we need to remember. And we can open the country for most of the economic activities and the school system. This is a slide we picked up from the Economist recently, which shows the effect of vaccinations, and effect of vaccinations mainly to prevent deaths. And most of the vaccines that the WHO has selected are considered to be good enough for this purpose. You see that the countries on this side, the number of deaths is come down drastically, and they have been managed to prevent deaths to a large extent in those countries. But those countries where the vaccination rates have been very, very low and not successful so far, the deaths continue to be high. And this is a clear case where the vaccination actually works. And Sri Lankan vaccination has not been too bad. I mean, there were some hiccups in the beginning when probably we did not adhere to the vaccination deployment plan, but I think it was restored halfway through, and on the whole, Sri Lanka has been able to vaccinate a large segment of the population over 30 years. I picked up this slide. This is Sun Tzu, one of a famous warrior, military leader of the 5th century, I believe. I just want to highlight two things. Just like in war, in pandemics like what we are going through now, two things are crucially important. One is the way we time our activities and how we maintain the momentum. It gives the parable of falcon striking the body of a prey. It is because of the timing, whereas torrential waters toss boulders around. That is because of the momentum. And water shapes its course according to the nature of the ground. And in our fight against COVID-19 and any pandemic, similar pandemic, we need to actually make sure that our timing is right and we maintain the momentum in our response. And these are the main elements in the response that we saw in the last 1.5 years. We have prevention and knowledge of management, including data and analytics, very important. We have the administration and governance. We have the clinical management services, surveillance on one side, laboratory services, and all this we say are responsible or crucially important to maintain what is the homeostasis of the whole system. And for each of them must be timely and should have momentum. But in some instances, whether you might not agree with me entirely, but we found that in many instances, our systems or units that are important in the fight against COVID sometimes tend to function in silos. The other difficulty we saw was that the quality of integrated real-time data for analysis to generate evidence-based decisions was not forthcoming in a timely way. We had strong leadership overall at the top, but I think at different points, there have been weakness gaps from time to time in different segments. And therefore, we had a number of coordination issues at lower levels. And of course, lack of a strategic vision and the lack of a cohesive information dissemination and public engagement process. I think this communication with the public and engagement with the public probably stood out as a weak area in the entire response that we saw. And why should we keep on being alert? We have to be alert, and we continue to be alert because this famous saying by René Dubos, who was the person who coined the expression, think globally, act locally. And he said, human destiny is bound to remain a gamble because at an unpredictable time and in some unforeseeable manner, nature will strike back. So we have to expect another pandemic sooner or later, and we should be totally prepared. And what does that mean? That means we need to have a strong health system to make Sri Lanka a better equipped country to face such challenges, natural disasters, pandemics, and any other serious health threats. What would it take? It would take good governance and coordination. It would take good disaster preparedness and response. By this, I propose that we need to develop a CDC, the Centers of Disease Control type of infection control system, which you find in a number of other countries, even regionally. And we need better reliance on digital technologies and data management. We need to continue to strengthen our PHC system, which is going to be the backbone of any fight against this type of pandemic. We need to address HR issues that are contained in it. And very importantly, we have to improve our risk communication and community engagement. Ultimately, we need to adopt a One Health approach. This is my concept of a Center for Disease Control-like organization of Sri Lanka. Under one roof, we need surveillance and epidemiology. We then need emergency preparedness and response to come under that. And we need laboratory service. Some of my colleagues might say, and some of the public health programs also here. But this has to be supported and backed by an extremely efficient, well-coordinated, integrated, real-time data system, which can do all the analytics. And these are some of the functions of such a CDC type system. They have to be on the cutting edge of health security, put science into action, fight diseases before they reach our borders, and in general, nurture public health. Now I'm going to finish. The summary of the key lessons and challenges I would summarize like this. Epidemiology is dynamic, uncertain due to a lack of data, driven by variable public health responses and complicated by variants of concern. Healthcare systems and workers under extreme pressure in terms of capacity. Surveillance systems finding it hard to cope with the high force of infection. Communities have experienced an erosion of social cohesion, access to education and income. Public health and social measures have considerable social and economic costs and must be risk-based, regularly reviewed on the basis of timely public health intelligence. The infodemic of misinformation, which was obviously seen so many times during the epidemic, must be countered by credible information. And the vaccination program, which is moving effectively, has to be continued to a finish. Ultimately, we need comprehensive preparedness and emergency response systems in this country. Because the costs of effective preparedness are dwarfed by the costs of failure to prepare. We need to remember that. And finally, I go back to Sun Tzu, who said, every battle is won before it is fought. Good preparation. Thank you very much. That's it.
We are a little behind time, but I've checked with the organizers. We can have a few questions. I saw several questions on the chat, but actually you have the opportunity of unmuting yourself and asking the questions verbally. So I will give you that chance now for people who want to ask a question verbally from any particular speaker. You could do so now. I hope the host has allowed them to unmute and speak.
I am Dr. Dilini Nakkavita.
Yes, go ahead, Dilini.
So this question is for Dr. Thushan and Dr. Chandima. So what is the recommendation for the third dose for high-risk groups?
Chandima, would you like to respond first and then I'll ask Thushan to respond?
Well, so we need to consider our local context. So as Professor Rohini pointed out, majority of our people has received the Sinopharm vaccination, especially the older people, and majority of the high risk group has received the Sinopharm vaccination as their primary vaccine. So in deciding of the third dose or a booster dose for those, especially the high risk groups, has to be determined based on our data. So we saw that the ones who received the Sinopharm vaccination, especially the ones who are above the age of 60, seems to be producing less antibodies compared to the others, and it is very evident with protective neutralizing antibodies. And our data also showed even the ones, the AstraZeneca vaccine recipients, the healthcare workers, at 6 months, there is a significant reduction of the antibodies, even after 2 doses, especially with regard to the current circulating variant in Sri Lanka. So all these has to be taken into account when deciding a booster dose in Sri Lanka, and especially for the ones who are immunocompromised. My personal opinion would be, considering these facts, the booster dose would be, needed for those special groups, but not for everyone in general, because, even the healthcare workers at 6 months, we did the T cell and B cell memory, cell responses. And we clearly saw very good memory T and B cell responses among healthcare workers even at 6 months. And there were some individuals even with one single dose at 6 months, there were good memory cell responses, especially among the younger population with regard to Covishield vaccine. So this has to be considered, and especially the high risk groups, it's worth considering a additional dose. Especially the data just came out yesterday from Nature also gives some evidence to show that, with Delta, the immune escape from Delta is more evident at least in vitro models. So that those has to be considered in deciding third dose or a booster dose. I think Thushan, some may have.
Thushan, you can add to that if you.
Thank you. I'd agree with everything. I think the only thing I'd add to that is that we need more granularity when we talk about high-risk groups. And it's quite clear that in some individuals, like patients who've had transplants of various sorts, the primary course results in something like 20 to 30% seroconversion. And so in those sorts of individuals, I think we need to consider that a 3-dose course should be the primary and not consider this as a booster dose. But there are obviously other high-risk groups, badged like patients with chronic cardiac disease or respiratory disease who are younger, who may have had a good response. So I think we need more granularity, and the immunosuppressed patients that Chandima mentioned should be having a 3rd dose as a part of their normal course.
Okay, go ahead. Anyone else who
Thank
wish
you.
to ask a question can go ahead. I saw several other questions on the chat.
Yeah, again, regarding the intranasal vaccine. So is there a place of a intranasal nasal spray vaccine when considering the mucosal immunity to achieve the sterilizing immunization after IM vaccines? Or are there any clinical trials going on? Or
Well,
what is the place of that?
there are plenty of trials trying out with intranasal methods. So, as I mentioned in my slides also, the intranasal vaccine provides upper respiratory tract IgA levels quite significantly, which will definitely help in assisting the mucosal immunity as compared to the IM injections currently used right now in quite many parts of the world. So there are some vaccines in the last stages of clinical trials, so hopefully it will be available soon. And the sterilizing immunity is the one which is ideal from a vaccination program, which will help to prevent the spread of the infection, and also probably will help in preventing. So yes, nasal spray vaccines are still under clinical trial and they are not registered yet, and hopefully it will be registered soon, which will help to provide sterilizing immunity in the mucosal areas.
Thank you, Chandima.
Any further questions? I see a question to Professor Rohini Fernandopulle about remdesivir. I think you mentioned it. Have you seen it in the chat?
Yeah,
Rohini
I
asked.
think yes, that that's whether it's it has been sent for waiver of registration at the moment, but we are keeping it because now we are using this new mechanism where we are going to monitor it. So probably by next week, we will either give an emergency use permission or a waiver of registration, depending on what sort of documentation they have provided and if the committee agrees.
There was a question to Palitha Abeykoon, but I think he's not here. Is Palitha here? Palitha, are you here?
Yeah, I am back. Yeah, I'm here.
All right, Palitha, there was a question which was a very general question. So which asked, what do you recommend now?
To do what?
I don't know what he meant, what is the overall recommendation for the interventions now?
That's very difficult. I will try to do so many different things. But the question is with regard to what should Sri Lanka be doing in terms of the stringency levels that we want to apply from now on.
I think that's what he implied, yes.
Yeah. Maybe tomorrow they'll make a decision on that. I mean, there's nothing sort of black and white. We can say it's always very dicey and fluid to make these kinds of comments and projections and predictions. But the general impression after doing in fact, we just finished today, did the projections using all these different things which we are submitting to the government. If we carry on for another week like this, at least close to what stringency we have now, and combine that with the other public health measures that people have to adopt, which now they are getting used to, by about November, mid-November, December, our situation should improve a lot. On the other hand, if we continue at the same level of stringency for another 2 weeks, till about, say, the last part of September, in fact, one of the things we are looking for is whether we can move from the current red zone that some of these Western countries have imposed, and Sri Lanka is classified as a red country along with a few other countries, and trying to see whether you can get back to the amber. By mid-November or end November, we should be able to do that. That's not that's not a gold standard as such, but at least some people quote that as for travel and things like that. For example, now anyone going from Sri Lanka to UK right now, because we are a red country, can't go direct to the UK. They have to spend 10 days or something in a country which is amber or green. So many people I know are going to spend a week or 10 days in Serbia. Now recently Dubai has opened for Sri Lanka. Many people are going to Dubai to spend the 10 days and then go to UK. So all that hassle we can remove. That's not the most important thing is epidemic, but that is, I'm just giving one indicator, which is also useful for us, which means our cases will come down and the deaths will come down. So the situation in Sri Lanka will improve. But we need to maintain a little bit more vigilance in terms of stringency and strict observance of public health and social measures. But most of all, our vaccine, like all our speakers were saying this afternoon, our vaccine program is going quite well, and that should kick in. We should see some positive results maybe by the end of this month or next month in terms of its impact. So it's a long answer to the short question, but it's difficult to give a short answer.
Thank you, Palitha. There is a question now to the Thushan and Chaminda, whether checking for antibodies is necessary before deciding whether to give third dose.
Well, I wish I knew that I knew the answer for that question, because we seem to, especially the healthcare workers and doctors seem to be checking their antibody levels and going for a booster dose. So that is one of the key research questions, everyone is looking at, but that is one of the key, gap in our research. We don't know what level of antibodies are protective. And, we talk about neutralizing antibodies, the levels, and especially with upcoming variants. So still the current vaccine is, the backbone is from the wild type. So we don't know how effective would be having a third dose for normal, with healthy population. So as at today, we don't have a level as such to decide on a booster dose, but, hopefully future research will find an answer for this.
Thushan, do you want to add to that?
No, I agree. I would discourage from doing that at the moment because of the reasons Chandima highlighted, and also because, as he highlighted before, there are many other things other than antibodies that may provide protection against severe disease. So while, as we've heard, a lot of the world is, hasn't had a primary course, I think we shouldn't be doing this in healthy individuals to give booster doses.
Okay. Anyone else who wishes to ask a question? Yes, go ahead.
Yeah, this question to Professor Rohini Fernandopulle. First of all, I must thank her for being open regarding the issues NMRA had regarding vaccines and so on. My question is that there's a misunderstanding among public that ivermectin is a drug given to animals and it has been now recommended for humans. I can remember we did some trials with Professor Ismail, use of ivermectin for filariasis. There we found there's no added advantage in ivermectin over DEC. But in a lot of African countries, they use ivermectin to treat onchocerciasis type of filariasis. So even here, I think the vets use it for diarrhea treatment. So are there two different types of products imported to country for animal use and human use?
Thing is, I am not sure because I know it's brought in for the veterinary sector because we don't do registration of veterinary medicines in NMRA. So we, the ones that are now available for sale in the pharmacies are the ones that are for human use. It is developed for whatever the active pharmaceutical ingredient is probably the same, but I'm not sure about the excipients. So whatever is now available officially is for human use.
I think we have had a very successful session. We've had very clear explanations of very complex problems. It's difficult to summarize all that was said, but I think one conclusion we can take from here is the need for clear and accurate data in decision making, whether it be regarding vaccination or intervention. I think all the speakers have highlighted it, and they have been cautious in making recommendations where the available data is not there. I think that is the clearest message I got from the presentations of everybody, including Rohini's pharmaceutical regulation. I think I will not say anything more. We've had two very good presentations on the laboratory aspects in the sense, the value of sequencing and in decision making for both clinical decision making as well as public health decision making. And then we saw a clear example of how a public health physician used that information in making the decisions. And we had an input regarding the pharmaceutical regulation aspect when in an emergency, decisions have to be taken in the short time. I think we will wind up now with thanks to all 4 presenters who did an excellent job within the given time. Thank you very much. And I won't say good evening or it's good afternoon here. I don't know what it is in the other countries. Thank you. We will wind up now.